Dear student, today I am coming with a new topic, transcription in prokaryotes. Transcription is the process of synthesis of RNA from DNA. This is the second step of central dogma, which is known as transcription. The subsequent process of transcription is called translation. Let us see what is central dogma. Central dogma is the replication, transcription, and translation. The replication means the synthesis of DNA from DNA, that is called replication. The synthesis of RNA from DNA, that is called transcription, and synthesis of protein from RNA, that is called translation. The entire three process from replication of DNA to synthesis of protein is called central dogma of life. Let us see what is transcription. As I told in earlier slide, the transcription is the process of synthesis of RNA from DNA. This is controlled by DNA present in the nucleus in case of eukaryote cell. In prokaryote cell, as they do not have nucleus, this process is taking place in cytoplasm. How transcription processes start? Transcription process starts when RNA polymerase enzyme bind in the promoter site of DNA strand with the help of sigma factor. Sigma factor is required here to identify the promoter site in the DNA strands for the RNA polymerase enzyme. Transcription process is stopped when Rho factor bind in the DNA template strands and prevent the movement of RNA polymerase. There, or sometimes the polydendron sequence of RNA, that is the uniform base sequences present in the DNA template strands, also facilitate in termination of RNA synthesis. Let us see the structure of RNA polymerase enzyme. RNA polymerase enzyme having 4 subunits and 1 sigma factor. Until and unless binding this sigma factor with the RNA polymerase, that and RNA polymerase cannot initiate the transcription process. The core enzyme part is called RNA, that's 4 subunits and the sigma factor. Altogether, this enzyme complex is called holoenzyme. That is the figure of RNA polymerase of E. coli. Let us see the first step of transcription, that is initiation. In the DNA strand, there are two promoter sites. They are having base sequence. This is the prepno box and this is the sequence of TTGACA. This is towards the upstream about 10 base pair away from start of transcription, and this is about 35 bases away from start of transcription. This is the coding region of DNA taking part in transcription process. The binding of the enzyme RNA polymerase to DNA is the prerequisite for the transcription to start. As I told, the specific region on the DNA where the enzyme bind is known as promoter region. These two regions is the promoter regions where enzyme bind in the DNA strand. There are two basic sequences of the DNA strand which the sigma factor of RNA polymerase can recognize for initiation of transcription. These are two promoter sites. Any one site will recognize by the sigma factor. This is RNA polymerase 4 subunits. This is sigma factor. When this complex will form, then only RNA polymerase enzyme can select the or identify the region where transcription will start. The first process is called initiation, means transcription process will start in presence of sigma factor. After starting the process, there is a sigma factor have no function. It will release from the complex. After subsequent process is elongation. As a holoenzyme means the complex of sigma factor and RNA polymerase enzyme recognize the promoter region the sigma factor is released and transcription proceeds. RNA is synthesized from 5 to 3' direction, means this is the template stance, and we know that it can bind only in anti-parallel directions. If this is from this strand will in 3' direction and this will start from 5' direction. This way the RNA polymerase enzyme will move towards this side and the mRNA length of the mRNA will elongate. This is the process of elongation, and RNA polymerase enzyme utilize the triophosphates, ribonucleotide triphosphate, ATP, CTP, UTP for the formation of RNA. This is the elongation process of transcription. Termination. There are two types of termination. One is row dependent termination, another one is row independent termination. Row, this is the row factor. This row factor, at the time of terminations, row factor comes and bind on the DNA template stance, but not with the RNA polymerase enzyme. When the row factor bind on the DNA template stance, the RNA polymerase enzyme cannot move towards this side. At the same time, what will happen? If it cannot move, it cannot transcribe more. At the time, the RNA strand will segregate from the DNA strands and release as a new RNA. This one is called this termination is called row dependent termination. Another is their row independent termination. The termination in this case is brought about by the formation of hairpins of newly synthesized RNA. Here, newly synthesized hairpins means some odd structure, means that hairpins means cannot move the RNA polymerase enzymes. It cannot synthesize new RNA because RNA polymerase enzyme cannot move, means ultimately what happened? The transcription process will stop. This occurs due to the presence of palindromes, means same base sequence or base sequence having same read in opposite direction. All the sites having same base sequence. Suppose Xerox word having some kind of similarity in words. The rotor, these are having some kind of palindrome structure word. Such type of base sequence, when it will get, the process of transcription will stops. This is for this type of termination, row factor is not required. That is why it is called row independent termination. Let us see the mechanism of transcription process in animated form. This is the DNA template stance. This stance will work as a template, and this is the coding stance. This is the promoter site, this is the transcription unit, and this is the transcription site. Let us see the how process will start. First, RNA polymerase enzyme will bind with the sigma factor. This complex can identify the region where RNA transcription process will start, and they identify the region of DNA where promoter site is present. That when they will bind together, that transcription process will start. This is the DNA along with sigma factor. Transcription process will start. After starting the process, sigma factor will release from RNA polymerase enzyme, and subsequent process will carry out by only RNA polymerase enzyme. That next one is elongation. Next steps. Elongation means that when RNA polymerase enzymes moves towards this direction, from this to this direction, the elongation of RNA strand will start. Now this is also for the elongation. RNA polymerase enzyme move, and the newly synthesized RNA strand also increase. At the time afterwards, when I already told that row factor will bind in the DNA strand, means template strand, then what will happen? This RNA polymerase enzyme cannot move towards this site. Ultimately, what will happen? The process of transcription will stop. So this is called row dependent termination of transcription. When there will be no transcription, then what will happen? Newly synthesized RNA will release from the DNA strand, and it will move to the cytoplasm as a new mRNA or new RNA. The termination, newly synthesized RNA, and that these RNA polymerase enzyme again take part in the transcription process with this sigma factor, and a row again it will come and prevent the next RNA polymerase enzyme in transcription process. This is the mechanisms. You see this row factor again come and join in termination process. The RNA polymerase enzyme again go and take part with join with sigma factor and form the RNA polymerase and sigma complex, which is called holoenzyme. This was about mechanism of transcription in prokaryotic cell. I think that you have understand the process. I've made this most simplified way and animated. You just watch properly, listen again and again, and if you have any confusion, you may discuss with me. Thank you.